So, welcome everyone, and thank you for the kind introduction. I'm very pleased to share a short insight from my collaboration with ICNTS. Today, I'd like to take you on a short journey through how we structure and connect and evaluate large LC-MS datasets, turning a flood of raw signals into clear, reproducible information. The image you see in the background is Pragmatis australis, or common reed, represents our model system, a plant used in plant-based wastewater treatment that absorbs pollutants and acts like a living sensor. It's a perfect example of non-target screening, extracting meaning from patterns from a huge amount of complex data. In plant-based wastewater treatment systems, plants such as reed or duckweed, Limna minor, play an important role. They take up nutrients, but also trace organic compounds, pharmaceuticals, for example. We use these plants to simulate environmental processes under controlled laboratory conditions. In the lab, we study how these trace substances are metabolized and stored inside the plant tissue. For us, the plant are not the only subject to study. They are a rich source of chemical information. Each experiment produces a large data set that we can explore analytical, statistical, and through data integration. And that's really the heart of our work, using data integration to translate biological signals into quantitative understanding. This diagram shows how pharmaceuticals enter the water cycle through injection, in contact, or even washing. Many compounds such as diclofenac or carbamazepine are only partly degraded, even in plant-based wastewater treatment systems. They persist in the water and can accumulate in the environment, where they may trigger unexpected metabolic reactions in plants and animals. These reactions, new or altered metabolic pathways, are exactly what we are aiming to visualize through non-target screening. Challenge is that we don't know what to expect in advance, so we need systematic methods that help us explore the entire data space. Every study begins with a careful planning. At ICNTS, this is done by using design of experiments, a structured approach that allows us to vary several parameters systematically. We adjust key factors such as exposure time, pharmaceutical concentrations, solvent composition, and plant tissue type, keeping them as independent as possible. Before data collection, all sampling analytical steps are tested, optimized, and validated to make sure the workflow is stable. Plants are incubated under controlled conditions, then harvested and stored frozen to ensure reproducibility before further processing. Each sample then passes through a fully analytical chain like HPLC, ESI, TOF, and MS. The modular setup allows us already at the planning stage to define which parameters can later be statistically evaluated. It helps us clearly see the effects and understand them better. In the first analytical step, HPLC separates the extracts by polarity. This forms the basis of their individual retention times. Mass spectroscopy then determines the exact mass and isotope pattern for each compound. Together, they create a chemical fingerprint, hundreds or even thousands of features per sample. What matters most is the reproducibility. Parameters such as flow rate, gradient, gas flow, ionization, voltage were optimized in advance, again using DOE. This reduces noise, saves time, and ensures that the data remains stable and reliable for statistical analysis. After measurement, we have two large data sets, retention time from HPLC and mass value from MS. Often, again, thousands of signals per run. These datasets are processed and exported using the software MSAT-MINE, where alignment, gap filling, redundancy checks for adducts and isotopes ensure consistency. Afterwards, the data will be merged in an access database from Microsoft or with Python. Within the database, all signals are connected by retention time, embedded value, and abundance. The result is a clean, structured table in which signals receive a unique ID for every analytical run. The structure allows the data to be filtered later by parameters such as solvent, plant tissue, or treatment. This is exactly where my main focus lies, building the bridge between measurement and statistics. Before we move into statistics, we prepare the data carefully. We need to do some scaling like Pareto or unit variance, transforming the data, sometimes with log, and replacing missing values if needed. The goal is simple: make all signals comparable so that differences truly reflect biology and no measurement noise. Pre-processing is often underestimated, but it's what separates meaningful models from misleading ones. Only with clean, well-prepared data, we can drive reliable conclusions. Now, to the multivariate part. Principal component analysis, PCA, gives us an overview. Are the samples reproducible? Do we see clusters, patterns, or trends? It's just a starter for the analysis. Later on, OPLS-DA modeling then goes one step deeper, separating groups, for example, controlled versus treated samples. Each observation appears at a point in the score plot, while the corresponding variables or features are shown in the loading plot below. Because both score and loading plots are connected, we can easily see which variables drive the difference between the groups. This visual approach makes complex statistics intuitive and helps to reveal clear experimental signatures. In the next step, we explore the effects of different factors, like solvent type, plant tissue, exposure condition, and so on. Different extraction solvents already produce noticeable different metabolite profiles, so solvent choice is far from minor detail. Likewise, roots, leaves, and stems show distinct metabolic signatures. The variability can make interpretation more challenging, but it also enriches the dataset and gives a more complete picture of biological responses across multiple dimensions. The S-plot is a highlight, which connects statistics with chemistry and biology. It shows which variables or features mainly drive the separation between groups. Points located far on the left or right side of the S-curve are the most relevant markers. In our example, under diclofenac exposure, quercetin levels are increased, a typical stress metabolite. Such markers are identified using mass databases, isotope patterns, and adduct informations. But the real insight comes from the overall fingerprint, the complete pattern of correlated features, not from a single compound. It's this system-level response that tells us how the plant reacts on stress. At this point, it's worth pausing for a classical reminder. Correlation does not mean causation. With large datasets, the chance of random correlation increases. The famous example in the study from Oldenburg, where birth rates correlate with storks' populations. Of course, there was no real connection, but it still looked statistically significant. Or think of the cat on the roof analogy. Just because you see it sitting there doesn't mean it caused the damage. Such coincidence can easily lead to overfitted models just because of random chance correlations. So we keep in mind, absence of evidence is not evidence of absence. Or more simply, even statistics is not entirely safe from Murphy's Law. That brings us back to DoE, a tool that helps us move from simple correlation to real cause and effect understanding. Instead of changing one factor at a time, we design experiments that vary several factors simultaneously, independent, and systematically. Experiments always consume time and resources, and the budget is never big enough. So searching blindly, and by blindly I mean OFAD, for the needle in the haystack can't be the goal. Instead, if we start with a clear plan and a goal problem definition, we can design our experiments with purpose. If we are only interested in linear effects, we use simply fraction factorial designs, a limited but efficient set of experiments. When we want to study interactions, we can extend the design with a fold-over step, complementing our experimental set with previously excluded experiments, but only because we see the need for that. Including center points help us to check reproducibility and detect possible nonlinear effects. And if those nonlinear predictors turn out to be relevant, a few additional experiments per variable are usually enough to verify them. This is normally not part of multivariate data analysis because it most time just checks about linearity. This approach helps us to identify key features and boundaries of our investigation with much higher confidence. It also gives us a model that is robust, interpretable, and reproducible, essential for reliable environmental or process analysis. To sum up, non-target screening produces massive and complex data sets through systematic preparations and well-planned experiments. We can make these data sets both manageable and meaningful. Multivariate statistics help us discover patterns, trends, and clusters. Experimental design helps us to understand their causes. My contribution, together with ICNTS, is exactly at this intersection, connecting analytical chemistry, data management, and statistical design. The future of non-target screening in combination laboratory practice, statistics, and data science. When these disciplines work together, we move from a simple collecting data to truly understanding our system. For plant-based wastewater treatment, this means detecting containment earlier and managing the process more precisely. For us as researchers, it means better designed experiments and more reliable results. And as you have noticed, I've mentioned design of experiments quite a couple of times. This is no coincidence. It's one of my favorite topics. And if you found yourself thinking, I'd like to hear a little bit more about this DOE stuff, I'm happy to point you to our upcoming joint DOE training with ICNTS in November. Thank you very much for your attention. I'm looking forward. Maybe we can have a look at your data as well. Thank you very much.